field as well as the lab scientists you have now. You can have this lecture, and I think this is a very benefited, you will be all very benefited with this lecture. So after all, I want to request Dr. Vishnu to please take this session, and since this is a very basic thing we have very young students, so I think that should be started from the basic thing only. Thank you, Vishnu.
Thank you, Diva, for your nice words. So, students, I hope you are now, it's a last day of your this training, but I hope you are full of energy like the first day of your training. So, you say yes? Yes. So, that's good. Now, my topic for the presentation is development of molecular markers for the identification of important fungal genera. So, as topic says, development of molecular markers. So, we will see how we can develop the markers. In simple way, I will tell you the simple way of the development of molecular markers while going through this particular topic within this 1 hour. Okay. So, let us start with, so you know what is the marker. So, the topic is molecular marker, we should know what is marker. So, what you understand by marker? It is, you know, you all might be using marker pen, no? You are leveling your isolates while transferring, you used to write something that we use to mark. So, that is a marker. It can be that felt tip pen also. So, similarly, for the fungi also we have some those type of markers. Okay. So, what these are? In this case, it is something that serves to identify, predict or characterize the pathogen or fungi. So, what is molecular marker? It is a readily recognizable genetic trait. It can be gene, DNA fragment or gene product that used for identification, especially when there is a difficulty for the identification by other means of the identification. Okay. So, why these markers? Why we need to have one marker for the identification of fungi? So, during this training, and I know you are plant pathologists only, you know the importance of fungal taxonomy and importance of mycologists also. So, what they do, what we do there, we try to identify particular pathogen based on the some of the characteristics like these conidiophores characters, conidial characters, then that attachment we consider, and we consider size, shape also for the identification of particular species. Okay. So, further this identification through these morphological means is very important for the study of the evolutionary study also. So, but sometime what happens, this type of morphological characters they could not distinguish subspecies level sometime. For at that time we need to have some of the marker which can distinguish the species of that particular genera. So, for example, what happens, you know one pathogen Fusarium fusiculae, it causes Bakani disease of rice. You may not be aware of that much of this pathogen because it was a minor disease. Now, it is coming in a big extent, in particularly in this Indo-Gangetic plain where basmati varieties are grown. So, the pathogen is Fusarium fusiculae. So, how we used to identify that? If we go for morphology, you know for the Fusarium, you might be aware we have to see many characters starting with the chlamydospores, whether chlamydospore is present or not. Then you see it is not present, one you have gone one step further. Then you have seen macroconidia is present or not. If macroconidia is there, what are the shape of the conidia? Then what are the size of those conidia? Then you have gone further. Then you have seen the microconidia where they are present. Microconidia, that size of the microconidia also you have checked. Then afterwards you have gone for the arrangement of those microconidia. Then afterwards you find that it is arranged in chain. So, you came up to this level, yes, it is arranged in chain, it can be Fusarium fusiculae. But same time there is another pathogen which is having all these similar characteristics, Fusarium proliferatum. So, coming to this up to this level, now again you confused that whether it is Fusarium fusiculae or whether it is Fusarium proliferatum. Okay. So, in that case we need the role of the molecular characterization. Okay. Here comes the role of molecular these markers or molecular characterization. So, how, but you know this ITS, generally you are aware of this now, I hope. Then it is generally used for the characterization of the fungal pathogens. Okay. So, here in this case only, so you have identified this, this is, it can be Fusarium proliferatum or it can be Fusarium fusiculae. Afterwards you have taken ITS, these primers, you got to synthesize those primers, then afterwards you amplified that region. After amplification you have given it sequencing and you got the sequence. Now, then after getting sequence, what happens, you know two primers are used, left primer and forward or reverse primer we can say, or left or right primers. So, what happens, then you made the consensus. What happens by this, if it is this left primer, it is right primer, you have made contig where it covers both left and right region and common region. Now, your sequence is complete. Okay. Afterwards what you did, you have gone to the blast NCBI. Now, you are aware of NCBI, you have opened this NCBI, then blast N. So, nucleotide blast you are doing. Okay. Then afterwards you have pasted your sequence and you clicked it. Then what we used to check in NCBI, how we are identifying that this is a particular sequence and this is the species in NCBI. So, you have done all this, then you got number of hits where you got query coverage, what we used to see there, query coverage and percent identity. Okay. So, query coverage you could see 100%, 10 to 20 species, 100% query coverage. So, there Fusarium fusiculae, proliferatum, so many came, and identity 100%, 100%. So, then how you will identify? Then you again stuck in initial stage only, because it can't be all 10. So, how we have to identify it? So, this although we use molecular markers for the identification of fungi, but we need to use particular one, specific one which can distinguish particular species. So, you can't go that randomly, you can't take ITS and you can't go for the this sequencing of particular this species of the pathogen for the identification. Understood this much? So, further you know this taxonomy, this is very specialized subject also, we need that expertise for that also. Many people do not have this expertise for the identification. Then further I told you that this molecular markers, role of molecular markers came here. These are very robust and repeatable, and the nucleic acid based detection, it can be used in any developmental stage. So, if it is seed, you can use it, seedling you can use it, and it is in adult plant stage also you can use them, because pathogen is there, we are identifying based on the DNA of the pathogen. Okay. Then this sometime this molecular technology, molecular identification, it helps for the identification of unknown species also, and the application of these technologies in plant pathology, it can help for the study of the epidemiology and host pathogen interaction studies also. Okay. So, we have seen, I have given you one example of ITS. Further, when we sequence some sequence and we are going to blast and we are identifying on the basis of NCBI, then there are certain problems in NCBI also. Okay. So, if you go for NCBI blast search, there it has been advised that blast search identification against gene bank should be made with caution, as approximately 27% of these gene bank fungal ITS sequences were submitted with insufficient taxonomic, that insufficient that number of this identification. So, these are or not always whatever you are getting in blast search, that is not always correct. So, there may be insufficient identification also, and in addition about 20% of fungal sequences in gene bank, it can be incorrectly annotated also. And sometime what happens that there are many unpublished sequences. Sometime what happens, suppose I am not knowing much of this NCBI and sequence submission, I have sequenced, suppose this ITS I have sequenced what I was saying you. Then what I did, I tried to submit it in gene bank. Okay. Bank it, I have prepared login ID and I have followed the instruction what it is given there. Then afterwards what I found, they asked me some of the queries, I tried to give, but sometime I may not able to satisfy them. Then what they will do, they will publish, release it as an unpublished sequence. So, there are many unpublished sequence also in NCBI which generally leads to misidentification of the particular sequence. Okay. So, that is also a serious problem with this blast search. Then there third party annotation of records are not allowed, you know, and taxonomic names are not up to date due to the rapidly changing nature of the fungal taxonomy. Suppose in this case also this Bacane pathogen I was telling you, there are 3 species mainly responsible. So, one is I told you Fusiculae, proliferatum, one more is verticilloides. So, what happens when you search NCBI as a verticilloides, you can't get it. So, here its name is different, it is in the form of moniliformis. So, sometimes those information are not updated in NCBI also. So, that is also the problem, and most fungal species described thus far have not been sequenced and type strains are not obvious. You know, for the validation of authentic identification, your this sequence should be identical to the type strain. Okay. Sometimes those type strains are not available and many sequences are unnamed or only partially named. So, these are other problems in this, and then what we can do? We have seen there is a problem in taxonomy also, what we have seen morphological characterization, there are some problem with the this molecular characterization also. So, what we can do further? No? What is the solution for this? So, general solution to most of this would necessitate working with the mycologist. You need mycologist plus mycological characterization, यानी कि morphological characterization as well as molecular characterization, then only you can come up to some conclusion. Okay. So, then in order to find type strains, one needs to find out original publication of fungal species and search within reference sequence database is also there. Okay. Then however, this challenge is compounded by the fact that not all fungal species are available in the sequence reference sequence database. We have reference sequence database also, but all those reference sequences are not sequenced. Okay. So, you may not get all the sequences what you want to see there. Then there is the problem I told you, and there is some solution also. So, what is the need of molecular marker then? So, you can simply take ITS, you can sequence, you can go for morphological, you can use both also. So, then what is the need? Na? So, here some of the problem with the plant pathology is detection and identification of plant pathogen. These are essential for disease management, you know. If you you can't identify, you can't manage the disease also. So, this is the problem. Further, all disease management programs, they need a simple, safe and rapid, this cost effective method for the pathogen identification. That is also the need of the time. Then this conventional detection methods based on disease symptoms or laboratory identification, they can be used for the identification, but qualified person is needed. So, everyone can't identify particular fungi. Okay. So, further, these time consuming methods do not always provide unequivocal results. Further, we have different methods, but sometimes there is not uniform in your results. You won't get uniform results. So, we need something which is always, if we take it, if we use that, we will get that specific results it is needed. Okay. So, this further, now some pressure, and because of the globalization in agriculture, you know, there are transboundary movements also, and we need strict legislation also, and quarantine is the very it is having very important role. Then this for this phytosanitary inspections and quarantine regulations, these they demands this increased control measures not only between countries, but within the territories also. Then this selection of healthy and disease free propagation material to the prevention of spread of pathogen to another country, screening of mother plants, screening of plant tissue, you need to have some of the markers for those purpose. So, and we need to for some time some agrichemicals they develops resistance. To screening that also you need some of the molecular markers. By this you can trace the that particular strain and fungicide also. So, thus the development of methods capable to detect and identify pathogens in plant material is fast, accurate and sensitive manner. It is the need of the time. So, where we need the role of molecular markers in plant disease management and improving decision making regarding the use of fungicides and resistant cultivars also. Then the detection of mating and virulence type. You know, generally for some pathogens we have two mating types, plus and minus type. If both the populations are there, then chances of getting this mutation and virulence is more. So, to screen that also we need molecular markers. If molecular marker is there, you can run it with that particular your pathogen, and you can see whether positive type or negative type is present or not. Okay. Then further, for the virulence studies also, you need some of the pathogenicity related genes may be present in some of the isolates, they may absent in some. For that purpose also you need to have markers. Then monitoring crop exposure to pathogen inoculum and assessment of the pathogen population structure also you need to have markers. Then one of the biggest advantages of PCR based method is the diagnosis of the pre-symptomatic disease stage. Generally those morphological and other methods we use to diagnose it after the appearance of the symptoms. So, these methods are very sensitive and we can diagnose it in the pre-symptomatic stage also. So, generally these markers, if you classify markers, there are different types. They are phenotypic, you may all know. Biochemical markers are there. Then molecular markers. Molecular also sometimes we categorized based on immuno based markers and some are DNA based marker. So, here we are focusing on the DNA based markers for the detection of plant pathogens, and these DNA based markers are further divided into PCR based molecular markers and non PCR based molecular markers. So, yes, now we have understand that we have some problem with morphology, we have some problem with this molecular characterization also, then there is a need of some this rapid diagnosis, rapid and sensitive diagnosis. And now the what are the characteristic we should have in that particular diagnosis, that particular marker. So, it should be easily available first. It should, this method what marker you are developing, it should be easily available, and it is, we have seen rapid diagnosis is the need of the time. So, it should be very rapid in diagnosis, and it should be polymorphic and reproducible also. You should get same results wherever you apply, and this codominant inheritance and this recurrent occurrence in genome is also important, and this it should be very important, selectively neutral to environmental conditions for the management practices. So, you know this our agriculture, these crops are always in the stress, they are lots of management practices we used to apply, and further the variation is there in environmental conditions also. So, in all the environmental condition, it should able to amplify your pathogen. So, there is one example, you might be listening about the Panama wilt, that is very important disease, and there are, that is because of Fusarium oxysporum formispeciis cubans. So, what is there? There are 4 important races of this particular pathogen. Okay. Or what happened that race 1, these we have markers for race 1, race 2, 3, 4. So, Dita in the year 2010, she developed one marker which is specifically which can identify the race 4. TR4, you might have listening TR4, now it is a emerging threat in banana cultivation, you know. So, now few days back you might have listened one newspaper, this study also, what they are saying, it appeared in US, and what they are doing, they are deploying army there because it is soil borne pathogen. If anyone comes and if takes that soil material, it can be transferred to the other locality, other country. So, that much of importance of this disease is there. Now, it is appeared in almost 16 countries in including India also, one report is there. So, it was earlier, first it was reported in Australia, then it was not there in India also, it was not there, but that marker was there, that she has given one marker for the diagnosis of this particular this race. So, how we identified that this particular this race is present in the country? That is, we have used that marker, and based on that marker and reference strain, then it was identified that this particular race is present in the country. So, what it says that it was developed in some other country, but it is useful in other environment and other country also. So, you have to take care of that environmental, it should be stable in all the conditions. Then data exchange between different laboratories should be easy. So, and high genomic abundance, it should be that selected region should be in high genomic abundance, so that you will get good amplification because of that. So, coming to this PCR based molecular marker, you know these PCR based molecular markers are always preferable. Why? These are these are most diverse with many different strategies that use amplification of a nucleic acid target as a basic component. So, we have diverse platform in PCR also, and PCR also is among the fastest assay to yield results. So, it gives fastest results what is desirable for the marker. Then the specific nature of PCR reaction enables it to be used directly on specimen containing large amounts of host nucleic acid. If you have host nucleic acid and pathogen nucleic acid also, you can amplify your particular pathogen nucleic acid. So, that is the advantage of this PCR, and further you can use it, you can have quantitative information also in the form of real time PCR. You might be aware, I think already you have one lecture on that. So, we can have other modifications in the PCR also. So, that is why it is very important type of markers, and but this unfortunately a universal limitation of PCR for fungal diagnosis is the generally low amount of fungal DNA samples compared to viral or bacterial loads. Generally for the fungi will get low this DNA compared to other pathogens, that is the one disadvantage. But this limitation has driven in part the search of variation to enhance PCR sensitivity. So, there is the limitation, but there are some solutions also. So, one of the earliest PCR variation was the ability to see results in real time. So, one is you can quantify and you can see in this PCR form also, you know this qPCR, and then multiple probes can be employed to create multiplex reaction. So, in same time you can detect many pathogens, that is also the possibility in PCR. Then while the most common forms of PCR employ this thermocycling, we can get broad range of temperature in PCR. You know that from this 5 degree Celsius to 95 degree Celsius you can use. Okay. So, then yes, they are PCR based markers. So, now how we will develop that marker? We will see this, you know, the basic requirement for the this development of marker is the DNA. If you do not have DNA, you can't do further, you can't proceed further. Okay? So, DNA extraction, we should have suitable methodology for the extraction of DNA. Then further we have to, this is most critical, crucial and important step for the development of marker, that is the identification of appropriate target gene or genome fragment. So, that is the, then how you are identifying what is your target gene, based on only you will get specificity, sensitivity and you can validate further. So, this is most important step for the development of marker. Further, now you have DNA, you have identified particular region, particular this sequence for the marker development, you have designed primers, then what you will do? That further step is the DNA amplification, you will amplify it, then you will see whether, then validation. Here you have to first see, you have to, it is not like that you have selected, just you have taken one isolate of pathogen, you have designed primer and you are amplifying. So, that will not be correct, because why? Because in other isolates of this pathogen also that particular region should be present, then only you can have that particular marker for that particular pathogen. So, what is the need? So, you have to screen as many as possible isolates of the same species. Okay. So, it should be present in all, then only you can say yes, it is the diagnostic marker for that particular species. So, you have to screen large number of species and it should be specific to that particular species. You have to take some other species also, like if you are screening for the, suppose this Fusarium fusiculae only what we have started in started with. So, you need to screen with as many as possible Fusarium fusiculae. You need to have other rice pathogens also. How you eliminate the chances that it will not amplify the other pathogens of rice. So, in seed, suppose in rice seed there are many pathogens. So, you have to check all those pathogens and you have to further, it should not present in your host also. You have to take care of that host also, you have to check in host also whether that particular fragment is present or not. Okay. After checking all, you identified that yes, it is specific and it is present only in pathogen, suppose Fusarium fusiculae. Then what is the further step? We have to check the sensitivity. So, how much, what is the amount of DNA it can quantify. So, if it is not that much sensitive, further we can't utilize it further. So, if it is giving it is highly sensitive and it is highly specific, then we can further validate it in the symptomatic disease plants, in the cultures and in other soil and other parts, where in different parts of the plants also where there are chances of the presence of the pathogen is there. So, these are the steps for the development of molecular marker. So, this is the schematic this chart. It is showing this first step I have already told you, we need to have this good DNA extraction method. Simultaneously, we should have a specific target region. A specific target we can have through this your particular gene sequence also, and we can use some of other way for the identification of the specific targets. You can go for RAPD, you can go for URP, and you can identify a sequence, that band which is present in all the isolates. So, you can try through that for the development of the marker. So, marker we develop that way, we call it SCAR marker. So, by different means we have to select the sequence, then afterwards what you have to do, you have to amplify, then you have to validate. So, these are the important steps for the primer designing. So, you know, for DNA extraction methods, we should have suitable methodology to isolate nucleic acid from the enormous range of habitats that fungi can occupy are crucial for the molecular detection. Good methodology you should have. Okay. Then this critical to be successful isolation of nucleic acid are the methods used for extraction and sample preparation, as well as the sampling strategy employed. Then we have enormous variety of nucleic acid extraction method. We have manual methods and we have kits also, through which you can go for the this extraction of genomic DNA. Further, with unusual and difficult sample types, customized methods are typically employed. So, what are the key for the DNA extraction? We should have the good cell disruption or homogenization step in the presence of the appropriate buffer. So, homogenization and quality of the DNA matters. So, methods which provide an adequate level of cell disruption to enable satisfactory this nucleic acid extraction, but without too much nucleic acid damage or release of potential PCR inhibitors are required. So, in your nucleic acid you can have PCR inhibitors also that interfere the amplification of that particular your product. So, we should have method where we will have good nucleic acid and further we have less these PCR inhibitors and less damage to the cell also. So, I told you this designing, the second step is the designing of the PCR assay. So, what we do there, the process it is in 3 types. Suppose you have for this specific marker, you can take this, generally we can have 2 types of markers here. One is that is based on the genes which are used for identification, like conserved genes, ITS, IGS, beta tubulin, TAF, those can be used for the designing of the primers. And second type is the specific genes. For particular pathogen, there can be specific genes also. Like for Fusarium fusicula, you know, it produces gibberellic acid. So, specific gene, there is one cluster NRPS 31 that is only specific for that particular pathogen. So, you can use the specific particular gene for the development of primers. So, what, yes, you have decided, suppose you have decided that you have used this, you have to use ITS region, that conserved gene for the development of marker. Then what you do, you have to go for in silico analysis. Here also that particular region should be amplified, and through the some softwares like Mega, like BioEdit, you have to take number of isolates, as many as possible. Then afterwards you have to see the particular region which is only present in this your particular pathogen, suppose Fusarium fusiculae only, and not present in others. Okay. So afterwards what you identified, yes, it is present only in the this particular pathogen, suppose Fusarium fusiculae. Then what you have to do, next step is you have to design primers for that particular specific region of that pathogen. Okay. That is first we have to go for in silico analysis, then we have to afterwards we have to select the target gene, then we have to design the primers. So, these are the steps for the this designing assay. Then I told you this selection of the genes, we can select conserved as well as specific type of genes. This is ITS region, this ITS1 and ITS2, it has been used for development of molecular diagnostic for many fungal pathogens. In some cases what happens, the genetic variation within this genomic fragment is insufficient. So, I told you example of this earlier I told you this Fusarium fusiculae only, we are getting same hits, 100% identification, 100% query coverage, like this we are getting. So, it is not good for the development of marker for this ITS is not suitable for that particular pathogen. Okay. Then intraspecific and intra-individual variability also occurs in this particular region. Then second, which can be suitable for the development of marker is IGS region. It is intergenic spacer region, and this it shows the highest sequence variation in fungus genomes and can be useful when ITS regions have a low genetic variability. So, you can go for this IGS region also, and in fungi IGS has a length of this approximately 2 to 4 this kbp, which is a fairly long region for the primer design, and you can use it further. Then some problems with this IGS also, this IGS is more problematic for PCR amplification and DNA sequencing in comparison to ITS. Then we can have this ribosomal DNA genes like this 18S, 5.8S and 28S. So, you can target these for the development of markers also. It has been used as PCR targets for the detection of many fungi, and this type of DNA contains variable and conserved regions valuable for species level primer designing and identification, and they are these you can use further. This is some example where this how this marker based this detection, it helps the identification of the pathogen. You know, through phenotypic data only, if you can identify this 59% of the genus, through this you can in some cases with particular primers like ITS2, you can identify 81% of particular genus. Then apart from these ITS, IGS and ribosomal genes, you can use many other DNA regions have like I told you beta tubulin, then EF 1 alpha, that is elongation factor 1 alpha, and this calmodulin also you can use for the marker development. And this the beta tubulin gene has become this is also highly used for the development of marker, and this further you can take other specific genes. I have given you example like Fusarium fusiculae, you can take NRPS 31. Like that we have specific genes also for particular genera, for particular species, you can take those for the development of marker. So, this is the same, you can use the this mitochondrial DNA also, DNA genes, but sometime it can amplify plant these particular mitochondrial genes also, some drawbacks. So, you can use this RAPD profile where you are getting this unique sequence in all isolate, you can take that for the designing of the primers. And similarly like RAPD we have URP, that is more stable, that is URP is universal rice primers. So, it gives a similar type of bending pattern. So, you can use that also for the development of marker, and this for the identification of the unique target region. So, these are some of the examples where in case of wheat different types of markers are developed in different pathogens. You can see scar markers based on IGS, ITS, those regions they have designed primers and they are able to detect the pathogen. So, third step is this validation of the PCR assay. You have designed primers suppose based on some specific region. Now, you get amplified it in the this your particular pathogen, you have checked specificity, sensitivity also, then you have to validate in different in plant and in soils, in seeds, in different parts of the plant also, and in different environmental conditions. So, this you have to follow. Then this is one example, it is from this our laboratory where the scar marker is developed for the detection of Bipolaris sorokiniana causing spot blotch disease of wheat. You know spot blotch disease, it is nowadays it is one of the emerging disease also, and it is present this pathogen, it can survive in seed, it can attack seedlings also. Then that for this purpose one marker is developed from our laboratory. Here what they have done, this DNA extraction they have done from pathogen, wheat leaves and soil samples also, different parts of the plant and soil samples has taken. Afterwards PCR amplification, here random primers they have used as well as URP primers also they have used. Further they have cloned that particular unique region, and then develop these primers were designed for the development of markers, and then specificity and sensitivity test of the markers was tested. Further this detection of Bipolaris sorokiniana in inoculated plant sample in soil it has done. So, this is the you can see this is the unique fragment they have selected for the development of marker. Further this they have checked for number of the Bipolaris sorokiniana and they found it is only amplifying Bipolaris sorokiniana only. Further they have checked specificity and sensitivity of this particular primer, and these are the number of primers designed based on that unique region, and they identified one of them it can only amplify Bipolaris sorokiniana, not other pathogens, and this they have optimized the PCR condition for that particular diagnostics. So, another example I am giving you from starting this Fusarium fusarium example. There are many markers it is developed for the identification detection of this particular pathogen. PCR based markers are already there based on this translation elongation factor 1 alpha region, and this other some variation in that particular marker. TACMAN PCR real time PCR assay was also developed. So, what they did, they have used so many isolates of this particular pathogen, then they have designed probes, then they have seen that disease index in phenotypically, and they identified genotypically on this marker also can give similar results as per phenotypic data. Okay, so, it can be used for the evaluation of the resistance also, this particular marker with the help of real time PCR. Then I told you there are variations only I am telling you. One was that this TACMAN PCR, quantitative PCR, other is multiplex PCR. In this case, this in same time we can detect so many pathogens. Specific bands are there for specific pathogens. So, these are some of the examples where it is used. So, multiplex this PCR is used for simultaneous amplification of multiple target genes or targets or genes. Then nowadays much more variation, there are many technologies, many variations came in this marker development. So, now new is this PCR nucleic acid lateral flow immunoassay. So, in nucleic acid this lateral flow has developed. So, you can see if pathogen is present, then you can have 2 bands. If it is not there, then only 1 band is there. Then this multiplex tandem PCR is another variation in this PCR. Then some non-PCR based markers. Till now I was telling you for DNA markers we have PCR based and we have non-PCR based. So, till now I have told you these PCR based markers, then how you have to develop and how you can validate and what are the variations in this PCR based markers. So, quickly here also the main thing is same, you have to identify the unique region. Okay. So, that is the most important that point for the marker development. Here also you have to identify unique region, but you need not have this PCR for the amplification. Okay. And these are visible without going for the electrophoresis and others at the same time. So, this non-PCR molecular markers, this DNA sequencing, this sometimes all these non-PCR based or PCR based markers cost benefit ratio you have to take care. So, some of those non-PCR based marker, one is FISH. FISH is fluorescent in situ hybridization method. So, what is it? FISH is powerful method for the in situ detection of active growing organisms in environmental samples. So, this technique can visualize the precise location of particular DNA or RNA sequencing in the cytoplasm, organelles or nuclei of biological materials, and as a result the technique can detect metabolically active fungi directly in the environment without the extraction of the DNA also. So, the spatial distribution of growing mycelia on or within colonized substrata can also be investigated through this. So, this here also it is having 3 steps. The major step or the step of FISH involves the preparation of biological materials and then the labeling. We have fluorescent label or probes, and then we have to hybridize, then you have to go for the visualization. So, these are the steps for this. Then finally, you can this hybridization of fluorescence you can check with the help of microscope. Then this DNA array hybridization is the other methods. These are generally you know this macro array. One is micro array, other is a macro array. These are macro array based detection. This DNA array hybridization, it also known as reverse dot blot hybridization or macro array, is a technique based on hybridization of amplified and labeled genome regions of interest. This to immobilize oligonucleotides spotted on a solid support platform. So, you will get if present that particular your pathogen is present, you will get particular this type of coloration or blot type, so that you can visualize this and you can say yes, this particular pathogen is present in present there. So, this is another technique. So, this is description of this. I think I do not have more time, so I am going quickly. Then further some isothermal systems are also there. Then isothermal system, they do not require thermocycler to produce rapid temperature changes, but require only a simple platform such as heating blocks or water bath, dry bath you can use for this type of system. So, some of the example of this isothermal systems are this isothermal system, this you have rolling cycle amplification, RCA, lamp. Nowadays, it is very common for the detection of the plant these pathogens. This is loop mediated isothermal amplification. Also, the other variation you can use for the development of marker, and these are the details of the lamp. So, here some specific polymerase we use, that is BST polymerase, and we have different set of primers. So, we have inner and we have 2 inner and 2 outer primers in this case for the. So, specific what will happen? You are increasing the number of primers. So, what is the benefit of this? Your specificity will be increased. ठीक है. So we have multiple primers here and it is specific for the detection. Then another is recombinase polymerase amplification. This RPA, it is introduced in 2006 and now it is now one paper I could see where in case of fungi also people have used for the development of this diagnostics using this technology. So here also it has 2 primers and 1 probe. So here also specificity increased and visual detection is possible. So one example, I am from starting, I am going through this Fusarium fusiculae, then final conclusive what I want to show. This whole genome is produced. Based on this, I told you that we can select marker, it can be specific or it can be conjured. So based on whole genome analysis, we identified one region, that is NRPS 31, it is highly specific for the Fusarium fusiculae. Then we have checked specificity and sensitivity also. Further it is validated in field disease samples and soils. Then this is PCR based marker we developed. Further we modified it and we can we developed this RT-PCR based primers. Then which can be used for the this characterization of host resistance also. So further modification we could bring here is the we developed the LAMP based assay for the detection of this particular pathogen. These are the primers selected, list of primers. So you know this I told you these are we can visual detection is possible. So if pathogen is there, there is blue coloration it comes, if it is not there this purple. So we could validate it, specificity here also you have to check specificity, sensitivity and you have to validate your particular primer. So this PCR based what were positive in PCR based primers what could detect, it could detect it by this real time and LAMP based also, but more benefits of those advancement real time we can go for the resistant characterization also, LAMP we do not need PCR also. So these are the this is the one example what I have started and this is the conclusion. And conclusion of this particular topic is, you know, you have seen numerous methods are available for the this detection of plant pathogens. Regardless of the approach, important questions need to be answered prior to their inclusion into experiments are sensitivity, accuracy, robustness, frequency of testing and cost. All are important, you have to take care of all. Then despite many novel technologies being available, challenges remain to identify as unculturable fungi. If it is possible, you have to see and it should be useful in different environmental conditions. There is always a need to characterize fungi quickly and accurately. So that is why markers are needed always. Then next generation sequencing, these approaches will also provide promising ways of enlarging the scope of molecular detection. These studies will get some novel reasons also. So we can have more rapid and more sensitive detection methods. So that is all. Thank you all for your patience hearing. They understood all.
Any markers for Isotonius raniformis species जैसा कि in maize crop
Marker.
for anastomosis grouping.
Anastomosis grouping, there are some markers, but we have tested they are not working in all the conditions. So we have some initiative on that also. Now we have AG1A specific marker. Rhizoctonia solini, not for Sasaki. Understood?
Madam, what is the difference between marker and primer?
Primer. primer can be anything. Huh? Primer, this marker can be primer, but primer can't be marker. Huh? Primer, it can be for some genes, if you want to identify pathogenicity gene, you can have primer. If you want to amplify ITS region, you are having primer, but that primer is not a marker. Marker is for the specific diagnostic purpose. So, for the identification purpose only we have marker. Okay.
So thank you, Bishnu, for this excellent talk. And if you have some more question, madam is sitting in room number 20, Division of Plant Pathology, so go and discuss. And you can also meet to ma'am, because the age group she can tell you better. So I just want to give a and the file to madam.
It's not needed. Thank you. Thank you.
So thank you, Vishnu.
Thank you, Deva. So, students, you are in this very, what I will say, in the stage where you have to, every minute of your time, this is important. Huh? So, give time, work hard as much as possible, give your 100%, and these are crucial time for you, MSc, PhD. You have different type of load, and you have different type of problems are also there. So, you have some pressure, family pressure, you have to settle down also, job pressure is also there, but whatever you do, you give your 100%. Okay. Thank you.